The overall goal of this method is to quickly and affordably quantitate multiple biomarkers in cerebrospinal fluid, or CSF, to assess their use towards diagnosis and treatment monitoring.
This is a new assay that allows us to measure biomarkers that were not possible to measure before. And we think this can be used for diagnostic purposes and also to differentiate between different neurodegenerative diseases and potentially also give prognostic information.
The main advantage of this technique is that it's designed to be rapid, simple in sample preparation, highly specific and cost-effective for translation into the clinical lab.
The implications of this technique extend toward therapy or diagnosis, as this high-throughput test can be used in future clinical trials for novel therapies in neurodegeneration.
This methodology can also be applied to other diseases and tissues, such as urine for kidney disorders, plasma for cardiomyopathy, and cells for pluripotency.
To begin this procedure, resuspend the desired synthetic peptides to a 1 mg per milliliter stock concentration according to the manufacturer's instructions. Prepare 1 in 10 dilutions of the peptide from the stock concentration and pool 1,000 picomoles of each peptide into a low-binding microcentrifuge tube. Dry the pooled peptide sample in a speed vac concentrator. When finished, store the sample at -20 degrees Celsius. Next, aliquot 100 microliters of CSF into low-binding microcentrifuge tubes. Freeze-dry the CSF samples. Resuspend an aliquot of the pooled peptide sample in digestion buffer to obtain concentrations of 10 and 1 picomole per